Hi, I'm Xinyu from Sharif's lab. Today I'd like to introduce my project, recently published in the Journal of Neuroscience. Modulation of SK channels via calcium buffering tunes the intrinsic excitability of parvalbumin-expressing interneurons in neuropathic pain. Neuropathic pain is a chronic, debilitating disease that can be induced by nerve injury or occurs spontaneously in the somatosensory system. Common symptoms include mechanical allodynia, where a gentle touch evokes pain. It affects around 10% of the population worldwide, severely reducing the patient's quality of life and becoming a significant economic burden to individuals and to the society. Current pain management strategies are limited by their moderate efficacy and adverse side effects. This is due to our incomplete understanding of the pathological mechanisms underlying neuropathic pain. This slide shows one neural circuit for mechanical allodynia located in the dorsal horn of the spinal cord. In normal condition, a mechanical stimulus such as gentle touch gets transduced by touch-sensitive primary afferent fibers and activates two subgroups of interneurons, the inhibitory parvalbumin-expressing interneuron in red, or PV neurons, and excitatory interneurons expressing PKC gamma in green. The PV neurons inhibit their targets, such as the PKC gamma neurons, and thus suppress the activation of this pain-producing pathway. Following nerve injury, such as chronic constriction injury or CCI, the inhibitory output of PV neurons is reduced, which allows the activation of PKC gamma neurons and the production of mechanical allodynia. So far, the mechanisms underlying these disinhibitions of PV neurons remains to be fully understood. In this project, I examined the possible changes in the intrinsic excitability of PV neurons after nerve injury. Our team recorded the electrical activity of PV neurons in spinal cord slice preparation from naive and nerve-injured mice. I analyzed all the electrical features using the toolbox that I create called ElecFX. This is a MATLAB-based toolbox with graphical user interface. As presented by the sample recording, following nerve injury, PV neurons show stronger spike frequency adaptation, or SFA. The discharge duration became smaller and the frequency decay rate was faster. I also observed a stronger hyperpolarized voltage change in PV neurons of nerve-injured mice. This feature, so-called medium after hyperpolarization potential, or MHP, is in part mediated by small conductance calcium-activated potassium channels, also known as SK channels. The larger area and amplitude of MHP in CCI conditions suggested the hyperactivation of SK channels. Next, I investigated the ionic mechanisms underlying the observed changes in the excitability of PV neurons. I developed a Hodgkin-Huxley type model consisting of 6 types of ionic currents. The model also describes the dynamics of intracellular calcium, and its changing rate, Fi, depends on the concentration of intracellular calcium buffers. The model was fitted to the experimental recording from naive PV neurons. To be specific, we matched the voltage traces and the action potential cycles between the data and the model. To identify the minimal ionic changes that can induce the frequency adaptation we observed after nerve injury, I examined the contribution of each ion channel conductance to the SFA-associated properties. A decrease in the sodium currents or an increase in the leak currents drastically shut down spiking, but they also largely reduce the peak of the action potential. A 300% increase or decrease to zero of all 3 potassium channels did not lead to the change comparable to CCI condition. On the other hand, an increase in calcium currents did induce a strong SFA. Plotting each ionic currents further revealed an increase in calcium conductance elevate both the calcium current influx and the SK currents efflux and results in SFA. More importantly, this change disappears when SK channels were blocked or when only SK channel conductance was increased. This result indicates that both an increase in intracellular calcium and in SK currents are necessary for generating SFA. The role of SK currents in generating SFA was verified using both experimental and computational approaches. We showed that the application of an SK channel activator, 1-EBIO, induced SFA in naive PV neurons. We also simulated the effect of 1-EBIO by increasing the SK channel sensitivity to calcium, and such changes consistently reproduce the SFA observed from CCI PV neurons. This result indicates that a hyperactivation of SK channels can contribute to the reduced excitability of PV neurons after nerve injury. I next examined whether this could happen under physiological condition. In fact, our model hypothesized one possible mechanism. By reducing the calcium buffer concentration, we were able to reproduce the transient firing pattern observed in PV neurons after nerve injury. The model further revealed that in low calcium buffer condition, there is a large and fast calcium influx through the depolarized membrane, which activates SK channels, and this increased hyperpolarizing power eventually ceased firing in PV neurons. The contribution of SK currents was then verified experimentally by blocking SK channels in PV neurons of CCI mice and computationally reducing SK conductance to zero in low calcium buffer model. These results showed that blocking SK currents can effectively rescue the tonic firing of PV neurons. I next examined how these predicted mechanisms affects the local circuits of PV neurons. To do this, I developed an in vivo-like neural circuit as in earlier slide. Under normal conditions, the inhibitory output of PV neurons sufficiently suppress the activity of PKC gamma neurons, irrespective of the firing rate of presynaptic A-beta fibers. Following a decrease in calcium buffer, PV neurons shows a stronger SFA, which impairs its inhibition of PKC gamma neurons. The firing rate of PKC gamma neurons increases significantly in the hypothesized nerve-injured condition, and such changes can potentially activate pathways linked to mechanical allodynia. In conclusion, we found that PV neurons in the nerve-injured condition showed reduced excitability. Combining computational and experimental approaches, we confirmed the significant contribution of SK channels and predicted a calcium-dependent mechanisms that modulate PV neurons' excitability after nerve injury. A depletion of cytosolic calcium buffer allows for the rapid intracellular calcium accumulation, which in turn activate SK channels and suppress spike generation. These predicted mechanisms potentially allows for the hyperactivation of pathways associated with mechanical allodynia. Our results pinpoint SK channels as potential therapeutic targets for treating neuropathic pain symptoms. I'd like to express my gratitude to Khadra and Sharif's lab for their support throughout this project. Thank you for your attention.